with our enzyme inhibition. So in all of these cases, what we're referring to is what's known as Michaelis-Menten kinetics. And your Michaelis-Menten kinetics is what generates an enzyme plot that you see on the right here. So this is your characteristic symbolism or your characteristic data plot for Michaelis-Menten kinetics. So if you see an enzyme curve along those lines, that means we're following the Michaelis-Menten system, which is different than what you see in Chapter 7. So just kind of be aware of that. So with this, we went through our standard kinetic diagram. We could look at and define Vmax and Km, and we could interpret that off of that kind of a graph. But then the question is, what happens if you have inhibition? How do those inhibitors affect the overall reaction? And so there's a variety of different mechanisms by which your inhibitor can affect the reaction. And that gets categorized technically into 4 things, but you get 3 main categories. Your competitive inhibition, where your inhibitor literally binds to the active site, which blocks the substrate from interacting. Right, so how does that affect your Vmax and Km? Right, you can reason your way through it. Ultimately, all you need to have is it memorized what's happening in those situations. Your Vmax will remain unchanged because what's happening at Vmax is you've maximized your substrate concentration. So if we have so much substrate concentration that there's effectively no inhibitor, we're going to run into the exact same Vmax line, right? Because it's as if there's no inhibition. So we'd get the exact same maximum velocity for that enzyme. Right? So with a competitive inhibitor, your Vmax does not change. However, your Km will change. So what the Km is looking at is the affinity for the substrate. Okay, and what ends up happening is your Km will increase. And that has to do with how the inhibitor affects the overall reaction that we've got up top. Okay? So it's where does that inhibitor affect? What rates does it change? What concentrations does it change? How does that affect everything else? Okay, so in the case of a competitive inhibitor, your Km will increase. You're non-competitive and you're uncompetitive. Okay, so you've got those summaries provided to you, so you need to know what they mean. Okay, so on uncompetitive, we're saying that you have an inhibitor binds to the enzyme substrate complex, but not to the free enzyme. A non-competitive, it binds at any time during the reaction. Again, not to the active site. So when you're uncompetitive and non-competitive, you're binding someplace other than the active site, and that causes a change in the structure to prevent substrate from binding to the active site. All right. Depending on when and how it binds, that changes either your Vmax or your Km. Okay. So ultimately, you're responsible for knowing those comparisons, and then how does that plot out? So if we look at our competitive inhibition, and we're going to draw a new line on first just our standard data curve. Okay. Well, if Vmax does not change, I would expect to end my curve at the exact same point. But we would also expect to start roughly at the same point. But Km increases. Well, how do we interpret Km off of an enzyme curve? Anybody? I know it's early. So we look at for Vmax. Our maximum velocity is somewhere above, okay, where our enzyme curve tails off. Okay, it's an asymptote if you want to get into it mathematically. So it's somewhere above that. Half of Vmax, so now half of that height, okay, our Km is now where that value intersects our enzyme curve. There's our Km. Okay, so that's our Km for our standard line. I guess I should have done that all in red. Okay. If we have a competitive inhibitor, what happens? Km has to increase. So where should the new Km be? To the left or to the right of that red Km? To the right. It needs to be to the right of it. So we're somewhere over here. Okay, so that's our new Km value. Okay, if we go up to our half Vmax, because our Vmax is the same, I now need to kind of connect those 3 points with that kind of hyperbolic line. Okay? So what you end up seeing is kind of a flattening almost. It's not quite a flattening because it'll still plateau, but that initial curve is going to flatten out a little bit. Okay? You'll have a less steep slope on it. It'll shift it to the right. Okay? What happens if we go through and look at the Lineweaver-Burke plot? Well, in our Lineweaver-Burke plot, you get all sorts of data coming out of this. So they've got this kind of nicely labeled for you. You've got the no inhibition. Okay? So you've got a standard plot from that. With that information, you can go through and interpret all sorts of stuff. Okay? Where it's going to cross your y-axis is your 1 over V max. Where it crosses the x-axis is your negative 1 over Km. So you can go through and determine what the Km is and what V max is graphically from the Lineweaver-Burke plot without having to do some approximations. Because remember, the issue that we run into with our standard enzyme rate comparison is that we're fitting to a curved line. And depending on where our data is, we may be able to predict where Vmax is, we may not. If the only section of this curve that you could see was here, where's Vmax? Okay, you can't really see it. Until you plot out enough data where you start to see the plateau, that's the only point where you'd be able to pick out Vmax. So if your experiment isn't perfectly set up, you won't be able to predict Vmax off of a rate versus concentration curve. Okay? That's where the Lineweaver-Burke plot comes in. Because then it doesn't matter how much data we have off of that curve. If I go through and plot it in that fashion, I can get a straight line, and I can extrapolate that straight line through my y-intercept and through my x-intercept to predict what Vmax and Km are. Okay? Much more reliably than if I'm trying to interpret off of the standard rate versus substrate concentration curve. Does that kind of make sense? Okay. So Lineweaver-Burke allows us to do a lot more extrapolation, is a lot more powerful and a lot more accurate. Okay. Our standard enzyme kinetic curve, we're really guessing at where Vmax is. Okay. And if we're lucky, we're close. Okay. If we're not lucky, we just guessed and we were completely wrong. Okay. So that doesn't help us out as much. Okay. To really get an accurate Vmax and Km off of our right curve, we need a lot of experimentation, a lot. Okay? So we spend 58 hours in the lab or we spend 2 hours in the lab. Most people prefer the 2-hour lab. Okay? What happens as we add that competitive inhibitor? Well, this is where it comes in handy to remember or to have memorized what your kinetic effect is. Your Vmax remains unchanged. So if we go through and look at our competitive inhibitor on that Lineweaver-Burke plot, we would expect it to cross the y-axis at the exact same point because Vmax has not changed. Okay, but what has changed? Km becomes bigger. Well, where do we interpret Km off of our curve? That's our x-intercept. And if Km becomes bigger, what happens to 1 over Km? It gets smaller. So what we end up doing is plotting really just 2 points. We can now see how that inhibitor affects the overall reaction. Okay, kind of makes sense? So it's a question of understanding what are your kinetic effects. Once you know what the kinetic effects are, how does that affect your graph? So there's a lot of graphical interpretation. Questions with the competitive inhibitor because it only gets worse.
Question, when you shifted the Km to the right, what did you say was occurring?
So what we're looking at is how do we, let's see, let me clear everything. Let's see if I can, let's erase that and that. Okay. So what we're looking at is a no inhibition curve right here. From this curve, we can interpret what Vmax is based on our y-intercept, and we can interpret from our x-intercept what Km is. So this is our x-intercept and our y-intercept. Okay. So that's with no inhibitor. So we're trying to see how does the inhibitor affect the reaction? Okay? Is it a competitive inhibition or is it one of these other options? Okay? So if it is a competitive inhibitor, what's going to happen to Vmax and Km? Vmax does not change. So if I'm looking at this as a competitive inhibitor, and I now want to draw on that competitive inhibitor line, okay, it should cross at the exact same y-intercept. Because what is the y-intercept? 1 over Vmax. If Vmax remains the same, it should cross at the exact same point. Okay, so let's draw that in red. So we have a point value right there. Okay, and we've got some point over on this side, and we've got some point over on this side. Okay, so what we're trying to do is just take this information, what we know the kinetic effects to be, to predict what should happen to our line. Okay? So the next part, what's the only other thing that we know changes or doesn't change with our competitive inhibitor? We really only have Vmax and Km. Km for competitive inhibition does what? It increases.
It increases, which means the affinity for the substrate is increased. But is that specifically just referring to the enzyme, or because the competitive inhibitor isn't really enzyme, it's like kind of separate. So I could see, I don't understand how Km increases if affinity for the substrate is still the same for the enzyme. It's just you've introduced a competitive inhibitor.
Which now changes the relationship between the substrate and the enzyme, because now your enzyme concentration effectively changes. So the relationship between your substrate and enzyme will change. Okay? And that kind of comes from what Km means. So Km,
Also changes your enzyme substrate complex.
Yeah, it changes a lot of stuff.
Yeah.
I'm going to regret doing that, but what we're looking at is Km can be expressed in this fashion, and there was another one, I forget what the other one was. But what we're looking at is K2, K minus 1, and K1. If we look at the competitive inhibitor, okay, what that's doing is preventing K1. It's preventing that rate.
Correct.
So if K1 changes, how does that change everything else?
Oh, I see. From the math, okay, I get that.
Okay, so it's strictly not changing, which is the other thing. So we're kind of doing kind of a hand-wavy argument in how that's affecting it. Okay. So I'm going to go through and erase all that stuff again. So that disappears, that disappears. Our Vmax remains unchanged, so we're going to cross at the exact same point. We now run into the issue of what happens with our Km. Well, if Km gets bigger, where should it cross the x-axis? Well, our x-intercept was at negative 1 over Km. If Km gets bigger, what happens to that number? It gets smaller. So I need to shift closer to that 0. Okay, so the second point I can drop in here would say somewhere in there. It doesn't really matter. Okay. Since we know we're plotting a straight line, there's our predicted curve for competitive inhibition. Okay. This assumes perfect data and all that beautiful stuff. Everything you're going to see in your online systems, online homework, or tests will be perfect data. If you move into a lab environment, then everything goes out the window and you get to deal with a whole separate can of worms. Okay? So what we're doing is based off of either fully understanding what's happening to the mathematics for each of these inhibitors. How does Vmax change? How does Km change? Okay? Or you accept that's how it's going to change. How does that affect your plot? Okay. What you will be expected to do both in this class and probably any test that you will see in the future for any kind of advanced degree is how does it affect the plot? Okay. What they're interested in is how do you interpret data once given to you. Okay. It's not how does this end up changing through, because that requires a much deeper understanding of both the mathematics and the chemistry. You don't need that. What they need to see is how do you interpret data once you've got it. Okay? Other questions with this one? So what happens if we now switch to excuse me, non-competitive inhibition. Well, in the non-competitive inhibition, what happens? Your Vmax decreases and your Km remains unchanged. So if we go through again on our standard enzyme curve, how does that go through and change this? Well, Vmax needs to decrease. Well, where's Vmax for the curve that's shown? For our red line, where is it? Okay, somewhere up here, right? Okay. In our non-competitive inhibition, Vmax needs to drop. So where should I put that new Vmax? Somewhere below that line. Okay? The specifics of that is irrelevant. Okay, but we're now somewhere below that. So that's our new Vmax. So if I'm going to look at this enzyme curve and I want to see its relationship to something that is not inhibited at all, what we're looking at is how does it affect the curve? Well, number one, our Vmax needs to drop. All right, so that means if I'm going to drop a point on here, we're looking at something out here. All right, and I'll still start at a 0. Next issue is that Km remains unchanged. Well, what was Km for our red curve? So we go half of Vmax for our red curve, and where that intersects, okay, is our Km. So that's our half Vmax. I'm a little bit below it, but that's close enough. Okay, where does the new Km value need to be? That exact same place. How does that end up changing our curve? Right, not drawn quite as nicely, but it usually means that you end up seeing the curve come up sharper and level off, okay, where they intersect at the exact same Km value.
So it's even more hyperbolic in shape.
Yeah, more hyperbolic in shape. Okay. What happens in our Michaelis-Menten kinetics, in our Lineweaver-Burke? Again, we want to shift into the Lineweaver-Burke because depending on how much data we've collected out of our experiment, we may not be able to see a clear relationship, okay, on how that changes. Because that all relies on being able to interpret Vmax. If we don't have the data to be able to interpret Vmax off that curve, the Michaelis-Menten plot is useless. We need the Lineweaver-Burke. So what ends up changing? Okay, it was nice of them to go through and use different colors on this. So let's go through and erase this. Okay. So there's our no inhibition line. Okay, no inhibitor. What happens in our plot? Well, Vmax decreases. Okay, so if Vmax, which was our y-intercept, decreases, or is related to our y-intercept, where should our new plot line cross? It's 1 over Vmax. If Vmax gets smaller, what happens? 1 over Vmax gets bigger, and where I cross is further up on the y-axis. Km remains unchanged. What does that mean?
Your x-intercept.
The x-intercept stays exactly the same. We can now draw, well, that was a pretty horrible line, but you get the idea. Okay? And we end up with our new plot out of our Lineweaver-Burke. So what you're expected to do is take that information of what happens to Vmax and Km under these different inhibitor conditions and see what the result is. How does that affect the plot? So if we clear everything off of that, that's what they're showing here. You'll notice that they also only show dotted lines, okay, on the left side of that y-axis. Why do they show dotted lines? It all has to be theoretical. Why? To the left of the 0 is negative. What did we plot on the x-axis? 1 over the substrate concentration. Can we ever have a negative substrate concentration? No. Okay, so this is strictly extrapolation based off of a straight line fit that we put on the rest of our data. Right, that's why they're showing that dotted line on that side. So we can extrapolate that information out and find it. Do we have to do that? No, we could still figure out Km by looking at the slope of the line. Okay? That's a lot more difficult, particularly if we add in the inhibitor, because look what happens to our slope. It now depends on the concentration of the inhibitor and on its binding affinity with the enzyme. Okay? So if we're just trying to figure out our Km's and Vmax's, extrapolating out to the x-axis makes the most sense. Okay? If we again switch our type of inhibitor, if we have an uncompetitive inhibitor, that means our inhibitor now binds to the enzyme substrate complex, but not the free enzyme. Okay? This is again going to change our dynamics on how the reaction works. Okay? The result is that both Vmax and Km decrease. Okay? And you can go back to and try and derive these two things and understand where they're coming from, which is what Victor was asking about earlier, looking at what we defined Km to be and what we define Vmax to be. Once we know what those are, how does the inhibitor affect those things? Right? And then you can see what would change about Vmax based on what's changing in each of those expressions. Right? I don't find that particularly easy. Right? You can do it, and if you want to, you can talk to me after class and we can go through and actually set that up. But really what you're responsible for is what do you do once you know that information. Okay? So that information is now both Vmax and Km decrease. Okay? If we again move to just our, I keep calling it a Michaelis-Menten graph, but it's just our rate versus substrate graph. What would we expect to change?
Both your X and Y intercept change.
Okay. Both Vmax and Km decrease. Okay. Well, where was Vmax for the red curve? That's our Vmax line. So if Vmax decreases, where does my new Vmax need to be? Lower than it somewhere. Okay. Km also decreases. I hate drawing Km on this curve. So if we go to our half Vmax, that's roughly our Km. So our Km decreases. So our Km value, new value, needs to be somewhere in here. That's where it crosses. And trying to connect these with our hyperbolic line, it's something along those lines. Okay? Not particularly easy to draw those on there. So we again want to go through and see what happens when we move to the Lineweaver-Burke plot. Okay, well, what's going to change? Both Vmax and Km decrease. If Vmax decreases, what happens to your y-intercept? Goes up. Okay, so I have to put something up here. Okay, if Km decreases, what happens to my x-intercept? Okay, Km decreases, 1 over Km gets bigger. So, good graph for me to have picked. My value then shows up further over here. We connect the lines out, right, drawn kind of horribly, but since Km and Vmax are changing proportionally, what ends up happening is you end up with parallel lines. Okay? That's what you visualize out of the curve. Okay, what's the question? Go ahead.
I'm confused on that x-intercept.
If Km decreases, 1 over a smaller number is a bigger number.
Yeah. Okay.
Yeah. It's easy to get it backwards.
Bigger.
So it's a bigger magnitude. It's still a negative value.
Yeah.
Okay? So the result for your uncompetitive inhibitor is that you end up seeing parallel lines. Okay? Parallel to your non-inhibition curve. Okay? The last one that your textbook kind of throws in there, but does not give you any kind of plots for, is if you have mixed non-competitive inhibition. This is a fun one, because your inhibitor binds at some place other than the active site, which is why we're in this non-competitive / uncompetitive situation, okay? Or rather, not competitive, okay? At any time during the reaction. And maybe it prevents the substrate from binding. Ultimately, what it does is prevents product from forming. Okay, so it can maybe prevent the substrate, maybe not. Well, what ends up happening? Km increases and Vmax decreases. how does that affect our plot? Okay. Your Vmax decreases, so if we take a look at this side, we're down lower. Okay, and Km increases, so we're looking at a Km that's further out. We just kind of extrapolate way out there. There's roughly our half. Okay, we end up with something along those lines. I think I drew that right. Okay. What happens on our other curve? Well, again, extrapolate that data out. If Vmax gets smaller, what happens to your y-intercept? Your y-intercept is 1 over Vmax. It gets bigger. Okay. If Km increases, Km gets bigger, what happens to your x-intercept? It gets to a smaller value, so closer to 0. What ends up happening? Okay, we got to plot something along those lines. This is different from all of the others because what we end up seeing is if we look on the negative side, we see an intersection on the, or we see a, it's not intersection, is it? Where two lines cross? Oh, it is intersection. Okay, it sounds weird. You see our two lines intersect, okay, in the leftmost quadrant of our plot. Okay, at least that's how the textbook references it. What's happening is we see that crossover in here. None of the other ones have that cross in that quadrant. Okay? They either intersect directly at our Km value, okay, or they intersect, or they don't intersect. Okay? So it's being able to recognize those patterns and see what happens out of that. So before we move into chapter 7, I want to take a look at the homework. Oh, of course. Oh, yeah, helps if I close out of that. Okay, let's see if I can actually get this into the window. So, based off these curves, and your now notes that you have that covers all of that, what type of inhibition happens in each of these? I actually don't remember. In all honesty, I have to cheat and look at my sheets. I remember what the second one is. The first one's going to be competitive because Vmax does not change. Okay, so our first one's competitive inhibition and our second one is a mixed. Okay. So in our first one, what happens? Again?
The Vmax is unchanged and the Km increases.
Vmax remains unchanged. Where's my cursor? There I am. And our Km, oh, come on. Sorry, I'm trying to do this carefully. Increases, we said, right? So, equations. That's a lot of stuff happening in those equations. Right? It can be easy to get bogged down in. I just thought of something while I was setting this up. There's another way to look at it. First equation. Is Km affected? How do you know that? In this first equation, is Km affected? Why no?
Because you're just adding it to whatever.
There's no multiplier. Nothing is changing. Second equation, what's affected? Km is absolutely affected. Is that the only thing affected? No, something else is getting affected. Okay. This is where it becomes tricky. Yes, you're looking at a substrate concentration. Ultimately, what we said is Vmax is what needs to change. How is the substrate concentration going to affect Vmax? You'd have to rearrange this equation to see that, but you would see that this multiplier would eventually make it in front of Vmax. Okay. But ultimately, two things are being affected here. And when we talk about our reactions, there's only two things that can be affected. Km or Vmax. This has two things affected, which means the This can't be the answer either. Next one. Only one thing is being affected by our inhibitor. What is that one thing that's being affected? Km. Based off the curve, we said the Km was the only thing affected. There's our answer. You don't have to do any algebra, you just say, oh, that's only one thing's affected. What happens with the other one? What things are affected? Km and Vmax. Remember, there are two types of inhibition that affect both of those, okay? Which does make this question a little bit more challenging. But look at your answer choices. In the first one, we have a factor affecting, but it's only one. In the last one, we have a factor affecting, but it's only affecting Km. In the second choice, what's happening? We have our inhibitor factor jumping in two places. Okay? Based on these equations, that is the only one that could possibly apply. Okay? So it's being able to interpret off the curve what things get affected, and now saying what equation best matches that. You do not need to know the algebra behind this. Okay? It's a question of interpreting the graph and saying what things are affected. What does that mean mathematically? Somehow that term needs to be affected by something. It needs to have a multiplier changing it. If it's not there, then that can't be a proper representation. Okay? This level of interpretation is again what you're expected to do on advanced tests. Okay? You're given some data, conclude something. Okay? And in most tests, guess what? They're multiple choice. Okay? So they're setting you up to give you the option to be able to exclude answers, not just find the correct answer. You need to be able to exclude answers. Okay? Questions on that? You'll notice that I got it wrong down here because I didn't actually answer that. I just wanted to see if my interpretation was right here. I was right. Okay? Just in case you saw the red X's. Okay. Where are we? So that, sorry, go ahead.
So can we conclude that any time Vmax decreases or increases, it reduces the rate at which the substrate is converted into product?
When it decreases, yes. When it increases, no. If anything, if it increases, it's going to speed the rate at which substrate turns into product. Because Vmax is your maximum rate, right? So if you increase the rate, it should speed that up. Okay. I promise we're almost done with this whole enzyme kinetic thing. Okay, Chapter 7 brings in now kind of the concept of mechanisms and what's happening within these. And of course, someone decided to invent a whole bunch of extra single-letter terms and then look at single-letter term ratios and call those new single-letter terms. And you get all these different symbols that are confusing and trying to organize, just like with Vmax and Km and all that fun stuff. Okay? But the reason we have to introduce a new aspect happens because in Chapter 7, we look at something known as allosteric effectors. Okay, what did allosteric refer to? We've already kind of talked about it. It looks at cooperative effects. We could compare it back to the induced fit model. Okay? Something binds, the substrate changes its structure, which now affects how it binds the next thing. So we're looking at some kind of cooperativity with that. And if you remember back when we first mentioned this between, say, hemoglobin and myoglobin, what changed about the enzyme curve? Not quite steep, has to do with shape.
Sigmoidal.
You're looking at
Oh, yeah.
sigmoidal versus hyperbolic. Okay, well, the Michaelis-Menten mechanism that we just looked at really only applies to a hyperbolic curve. It cannot predict the sigmoidal shape. Okay, it fails in that respect. Okay, That's what Chapter 7 is kind of trying to address. Thankfully, we don't get into too much of the equations and all the plots and all that fun stuff. We're kind of looking at it, but not to a large extent. So we're talking about our allosteric effectors. You get feedback, pathways. Those are some big kind of terms that get floated out there. You should make sure that you're aware of what those terms mean so that you can go through and apply them appropriately. Okay? The other one that drops in here is active versus inactive. I'm undoubtedly going to get them backwards, but I'm going to try and see if I can get it right. Active is referred to as R. Inactive is referred to T. Anybody remember? Did I get that right? It's on like one of the next slides. Okay. So we get this R and T concept coming out of this. We run into some interesting kind of effector examples. We can talk about phosphorylation and zymogens. So two kind of interesting little things that come out of it. And then we look at the organics of enzyme reactions, about freaking time if you ask me. So what's happening in organic chemistry, what types of reactions occur, how does that actually translate into what our proteins go through and do. Right? So allosterism really just means change in shape. Okay? So what we're talking about is the enzyme or our protein is changing shape, and that process of changing shape affects its reactivity. Okay? So our allosteric effectors can have either a positive effect or a negative effect. If it's a positive effect, you're looking at an activator. Your substrates are often considered activators, right? Though not always. Your negative effect would be an inhibitor. Okay? So it's inhibiting the reaction. Okay? So if we go through and take a look at what happens to our curve, if we have no activator or inactivator, we have this kind of sigmoidal shape coming out of it. Okay? As we add more inhibitor, we're going to slow the rate of our reaction down. Why should we slow the reaction if we have an inhibitor? Because we have an inhibitor. It inhibits the rate of the reaction. Okay? What happens with an activator? Well, it's going to speed the rate of the reaction, again, up to a point. So we end up getting a sharper line on it. Okay? So it's looking at those effects and how that comes out of your curve. One of the things that you might actually start to note as well is that our inhibitor kind of introduces more of a sigmoidal shape. As we add more activator, what ends up happening to our sigmoidal shape? It becomes more hyperbolic. Okay? So with the presence of an activator, we can potentially see our curve start to match Michaelis-Menten kinetics. There are two types of enzymes that get referenced. Okay? And this is now your V system and K systems. Okay? I know we're bringing in a bunch more new terms. These are technically not new terms. What is a V system? V stood for our velocity or our rate, how fast the reaction went. So what are our two systems? A V system is one in which our max rate is changed. Okay, our V system changes our max velocity. Our K systems change our Km. Okay. We do run into an issue. Km solely applied to our Michaelis-Menten kinetics, which meant a hyperbolic curve. Do we have a hyperbolic curve anymore? No. So we can't strictly use Km anymore because Km only applies to the hyperbolic curve. Okay? So the new term that comes out of your allosteric effectors is this K a half. Okay? Why do we call it K a half? When did you know you had Km? At half of V max. This new K value that they're talking about is found the exact same way that you would find Km off of a hyperbolic curve. So it's named after it. So it's looking at the same interpretation. It's not the same thing because of how it's derived, because you're looking at that sigmoidal shape as opposed to the hyperbolic shape. Okay? So our understanding of Km from our Michaelis-Menten kinetics, we can derive. We can say K2, what was it? K2 plus K minus 1 over K1. Okay? The Km in allosteric proteins is not derived that way. we can still draw some information about that general concept by looking at the substrate concentration at, again, 1/2 Vmax. And at 1/2 Vmax, we can say that substrate is now related to this equilibrium term, our K term, which we call K1/2 as opposed to Km. So we get a similar derivation of a value, but that value holds different context than it does to Km. So it kind of makes sense? So we use some similar terms and some entirely different terms, because that makes it easier to understand. Okay, so this is our Km becoming K1/2. So if we look at Michaelis-Menten on our right, we can go through and derive what our Km is based off of our hyperbolic curve, and we can do all that fun stuff. Okay, we can look at our half or our Vmax. We can go to 1/2 Vmax. Where it intersects, we now have a Km value. Okay? That Km value holds the same context from this derivation, from our Michaelis-Menten plot. Okay? We can't use that same understanding when we move to our sigmoidal curve. Okay? So we can't call it Km anymore. Okay? But we can derive a number similarly as we did here by going through and looking at our Vmax, looking at our 1/2 Vmax, where it intersects. That's now not Km, because Km only applies to the Michaelis-Menten hyperbolic shape. We call it K0.5, okay, because that's still holding some of the information on how we could theoretically derive our binding constant from Michaelis-Menten dynamics. Okay, kind of, sort of. Okay, I just want you to be aware that we can associate a number with both of those K values through the same type of interpretation off our graphs. But that number holds very different meaning between K1/2 and Km. Yes?
Are you no longer referencing your x-intercept, or are you still doing that?
So we can't go through and plot it according to the Lineweaver-Burke plot. The Lineweaver-Burke plot only comes from the hyperbolic shape.
Right.
We're at a sigmoidal shape.
Right.
All of that Michaelis-Menten stuff that we've done gets tossed out the window.
Right.
Okay, so we can't do that interpretation anymore. So, if we go into our types of allosteric enzymes, okay, so it's again this changing of shape within it. There end up being two mechanisms used to explain this added complication of what happens with your enzyme changing its structure. You get a concerted mechanism and you get a sequential mechanism. Depending on which mechanism you look at, you get slightly different pieces of information, which is why we have two theories. Okay, the concerted mechanism explains a lot of enzyme kinetics, but it doesn't explain this other chunk. Okay? The sequential method explains the other chunk, but then kind of falls apart in the other systems. Okay, so we have to have these two different understandings of it. Why do we have two different mechanisms? Because think of the vast variety of what our enzymes can potentially do. We really assume they all run under the exact same mechanism. Okay? It just doesn't make sense to have everything fall under one category. Okay? Both are concerned about this reference to an R and a T state. Okay? So R for relaxed and T for tight or taut. I have no idea where that referencing is coming from. I tried to look up the scientists and see if they were French or German and see if they're made sense in active versus inactive. And I don't see anything that's really obvious there. The relaxed form is your active form of the enzyme. Your taut form or your tight form is the inactive form. Okay, so there's these two forms of our enzyme depending on what we're looking at. Okay, we're going to be looking at different forms of these. And this is our inactive. Okay. What's that?
We're just saying it's kind of counterintuitive because you're saying when something's relaxed that it wouldn't do anything.
I thought that too when I first went through it.
So just the
Yeah.
opposite of what you would think.
Think back to organic chemistry when you had nomenclature and you're like, all these stupid rules, why do I have to know all these rules? At least you had rules. Okay. So if we look at our conservative mechanism, what we've got are these two forms. Your R form, your active form, binds the substrate tightly. Okay? And it will change the structure to favor more substrate binding. The T form, or our Taut form, does not bind our substrate, okay? But it does bind the inhibitor, okay? Which then prevents more substrate from binding. So what we're looking at is an equilibrium between these two states. Okay? How do we then define that equilibrium? So frustrating. They bring in this term L, okay, and this term C. The L term is effectively the balance between your taut and your relaxed state. So it's the balance of your inactive versus your active state. Okay? So typically what we see is that your L ratio is significantly larger towards your taut state. Okay, so we see very large L values because your taut state is the native state. It's the one that you're going to find everywhere. As an inhibitor binds, what ends up happening is you now remove some of the taut state. What happens to the equilibrium? If I remove some of the taut state by adding an inhibitor, the equilibrium between these two is now broken. What has to happen? Careful, if we remove T, it's going to shift to form more taut state. What happens to the R state? It becomes less common. And what happens? You see the enzyme reaction slow down because there's less active enzyme. So if an inhibitor binds, it removes the taut state. Okay? We now have to replace that taut state by losing some of our active enzyme. The effective result is that our enzyme reaction slows down. What happens if we look at the reverse? What happens if substrate binds to our active state? Then we have less active state, so what happens? The reaction shifts to form more active. What ends up happening? Your inactive concentration is reduced, and what happens to the speed of the reaction? It speeds up. So we end up getting a faster reaction. So it's looking at the balance between these two states and what does an inhibitor do versus an activator do, and changing each of those states and the balance of the equilibrium between those two. Okay? So we can look at this L value as our balance of inactive to active. Okay?
So we'll try to reach equilibrium, right?
It will still always try to reach equilibrium. And that equilibrium is centered on the relationship between the relaxed and the taut forms, okay, or active and inactive. I'm going to try and make sure I always phrase those in the same situation. Okay. The other thing that pops up is they, of course, have to add another variable. So the variable that they include is this C value. Okay. The C value is the ratio of the binding affinity for each of those states for their respective partners. Okay. So what we're looking at is Kr, which would be the binding affinity of the active state for its substrate versus Kt, the affinity for the inhibitor and our taut state. Okay? So that relationship, how well they bind the inhibitor versus the activator, is now also going to potentially change our reaction. And what we want to be able to do is see how would that change our curves in each of these cases. Okay, so we get a lot of curves here. What happens as L gets really big? Whoa, sorry. My screen is freaking out. Okay, so here's L again. It's our taut over our relaxed. If L is really, really big, what does that mean?
Like you have a lot of inactive state.
We have a lot of inactive state. What would we expect for our curve? I'm intentionally trying to hide it so you can't see it.
I can't can't see what's on the
No.
y-axis.
That's exactly, I don't want you to look at the y-axis, that's why I'm trying to hide it.
Reaction is slow.
Okay, it's going to slow down the reaction. What is that going to do to our shape of our curve? More sigmoidal or more hyperbolic? Sigmoidal. Because what's going to end up happening is I have a lot of that taut state. As I bind stuff in there to change it into the relaxed state, I'm going to start to see a much larger cooperative effect. So the bigger our L value, the more sigmoidal our shape. Because I have to get up to a lot higher substrate concentrations to actually get the enzyme to work. So I'm going to see kind of that flat line at the beginning before it starts to spike and level out. What happens if we have very small L value? Okay. Now what we're saying is we have no taut state. We only have the active enzyme. If we only have the active enzyme, it's going to become more hyperbolic. There won't be any cooperativity because there's nothing to cooperate. It's already in its purely active form. It's ready to bind right away. So if we go through and take a look at your curves, L is massive, you get a very sigmoidal shape. And I know that doesn't show up very cleanly on that. Okay? But you're going to have to shift with kind of a flat line at the beginning. You have to really up the substrate concentration to get enough of a shift across into that relaxed active form. If you have virtually no taut state, what happens to your L value? Your L value is going to get relatively small, and we're purely in the active state, which means as soon as there's substrate there, what happens? It binds to the enzyme. We immediately see a reaction. So we move to a more hyperbolic curve as opposed to the sigmoidal. Okay? It would be nice if we didn't have to invent these stupid extra terms to this, but that's kind of where it's coming from. Does that kind of make sense?
It's an increase in L is an increase in the inactive state.
An increase in L, because L is inactive to active. Okay? An increase in L means the inactive is higher. Okay? Which means you're going to have a sigmoidal shape. You will see that cooperativity. Okay? These two chapters are full of freaking ratios, and they all use different symbols.
What do you mean by cooperativity?
As more substrate binds, more substrate can bind.
Perpetuates itself.
Okay, so it kind of self-feeds. So if you think back to your myoglobin versus hemoglobin, in hemoglobin, one oxygen binds, which changes the enzyme structure so that another oxygen can bind easier. That second oxygen binds, which now changes the enzyme structure so that another oxygen can bind easier. So you see a sigmoidal shape because it goes flat. We don't really see a response until all of a sudden it's going to spike. Okay, and go up really, really, really fast because you have enough substrate in there to now actually kind of self-perpetuate and speed the reaction along.
So if it's hyperbolic, then it's already active and therefore there's no cooperativity?
There's no cooperativity because it's already in its active state. There's nothing to actually cause it to become more active. If we're already active, you can't make it go more active. It's already there. So C was our ratio of binding effectiveness between our relaxed versus our taut states. So that's R being relaxed state and S being substrate, just like we were looking at with our enzyme Michaelis-Menten system. All right, so what happens when C changes? So,
Looking at Q sub C.
what's that?
Go back to equilibrium. You have
It's
the
a
one form, and then when it changes, you look at Q sub C.
It's kind of a Q, yeah. But you're looking at a ratio of those binding affinities. Okay? So if we look at the situation, and I always have to think through this one really carefully, the bigger our C value, what does that mean?
The bigger the active.
Okay?
Yeah.
We're saying our binding affinity
And the active.
in our active state is now really, really, really strong. Okay? So in that curve, what happens? Okay? Bigger our C, we're now saying it's grabbing substrate right and left. Okay? Really strong affinity. Okay? Well, if it has a maximum affinity, how can we make it more maximum? Does that kind of make sense? If we have a massive affinity, that active site is just grabbing things. Can it get any faster? No. What happens to the curve?
It becomes more
it's going to become more hyperbolic.
Really? Because I
Right.
see the.
So you
oh,
got to
I
be
see.
careful. Yeah, they cross.
The
So
colors
the
are weird, purple and blue.
bigger your enzyme affinity, your relaxed affinity for the substrate, the more hyperbolic the curve. Okay? Because if it's grabbing stuff at a maximum ability, I can't make that any more maximum. Does it kind of make sense? Yeah.
So are C and L are just totally interchangeable though. You just switch your T and R values.
Can't you look at like proportion, like they're
they're
directly
pro-
proportional?
They're going to have a similar relationship. Your L value is your concentrations. Your C value is a binding affinity or related to your binding affinity. So they're measuring different things. Okay? Yes, they are related to each other, and so you can try and go through and do a comparison and set that up. I definitely have not thought through it enough to do that because I want to think about it as little as possible, because the more I think about it, the more confused I get. So I try to get it squared off where I'm happy with it, and then I can move on from there.
What it says in the chapter, I mean, just straight up, it says, as the L value increases, so the free T form is more highly favored, the shape becomes more sigmoidal. The value
Yep.
for C decreases, the higher the affinity between substrate and R form, and the shape also becomes more sigmoidal.
Yeah.
So it's in an inverse
They
relationship.
invert relate. But should it make sense for them to invert relate? L is taut to relaxed, C is relaxed to taut. So it does actually make sense that they should be an inverted related, that thing, because they are looking at inverse relationships. Okay?
I think it makes sense, because if R is like the active one, then it's gonna be hyperbolic because it's already at its max, so it can't be sigmoidal because there's no cooperative binding, so.
Yeah.
You just gotta know which value you're doing.
Yeah. And so that's going to be the thing, is you're going to be asked L's and C's and all those different things. Yeah, don't even touch ice charts. I don't even want to look at that. So the textbook gives you kind of a nicer picture of this. So where we've been focusing and just kind of been talking off that it exists, we've been focusing just here. Okay, the balance between your relaxed and your taut forms. Okay, but as we were talking about, what happens as you change, so your K values are ultimately looking at this reaction. Oops, sorry, your K values are here in blue, officially kind of over here. And actually not that one, it's just your substrate. Okay, that's your balance of your K values in blue. The balance, which is your C. Okay, so C is in blue. Your L, it is L, right? Your L value is the balance in red. Okay, so they are of course going to be related because the L value is looking at relationship of R to T. Well, what is C looking at? T to its binding affinity. Well, if I change C, is that going to change the concentration of T?
Yes.
Is that going to change L?
Yes.
Yes. So they are all tied to each other, but they're looking at different aspects of the same overall picture. Okay? And so the balance in this concerted mechanism is that as we bind, we're now causing a shift in our balance between our relaxed and our taut forms, which is then changing the activity of our enzyme. Okay? So once I bind something to, say, the R form, the concentration of the R form now drops. Because its concentration drops, T has to change into R to replace its concentration. So you're doing Le Chatelier kinetics all the way through this.
So you're saying C is just concerned with T in this case?
No, C is the balance, so it's in blue. Whoops, shoot, don't do that.
Because it's going to be the same.
C is the
R
balance
itself.
here and over here.
Yeah.
Okay.
So C takes into account both those blue marks.
The binding affinity?
Yeah. Okay. L just takes into account the concentrations of R and T. Okay. So they are all absolutely going to be related to each other, and you're going to see those kind of inverse relationships because of what you're comparing in each of those states. Okay? Questions?
Is there a relationship between those, like if L is high, I don't know, can you get like?
So, well, that was one of the things that I think the textbook at least put out in a sort of succinct sentence, that if L goes big, C goes small, they're going to be inversely related.
Always?
Yeah,
Okay.
they should always be related like that. Okay. The secondary mechanism we can look at is the sequential allosteric enzymes. So we still have this taut and relaxed form, but it's now within the same enzyme. So there's two halves effectively to our enzyme, okay, as opposed to two separate concentrations that are now within the same thing. And our enzyme will exist always in that taut form. As substrate binds, we see a change in how the enzyme organizes its structure. Okay? Excuse me. Once the substrate binds, it now changes the conformation from our taut form, our inactive form, to our active form. Now it can actually potentially run its reaction. But in that same time that it changes to the active form, it also now affects the other half of the molecule. That other half of the molecule then changes your taut form into the active form. We now have two available binding sites for our substrate, and we can turn over faster. What happens if we have an inhibitor? Well, our inhibitor binds, okay, and changes our structure so that it makes it even more difficult for substrate to bind in our taut form. Okay? That makes it even more difficult for substrate to bind in the other side. But it also increases the affinity for the substrate. Or not substrate, what's that word? Inhibitor. Okay. So as you bind more substrate, it becomes slowly more active. As you bind inhibitor, it becomes slowly more inactive, okay, or inhibited. Okay, that's what your sequential enzyme is getting at. The reason why this was developed is that you get this big idea of cooperativity, and not just positive cooperativity, but negative cooperativity. Okay? As inhibitor binds, you get more inhibition. Okay? Whereas in our previous mechanism, we don't have a way of explaining that. It doesn't increase the affinity for the inhibitor. Okay? So that's our big kind of lesson out of the sequential allosteric enzymes, or the sequential mechanism. Okay? This might also make a little more sense because you see a little bit more of this induced fit model behind it, whereas the other one's more kind of a lock and key system. Oh, please, can I be done with it? Okay. So you're really just trying to evaluate those interpretations. This section is going to be brutal because you have to make sure you try and tie all those things and keep them, all these similar concepts that are different, sort of separated from each other. Okay. As far...
Lock and key is cooperativity then, basically, when it like just fits automatically?
It can be... Well, no. So induced fit is really where you see your negative cooperativity. That's the big aspect of the sequential mechanism. For the concerted mechanism, we don't see a negative cooperativity. Once our inhibitor binds, we only have taut state. It doesn't further become less. It doesn't further become, less active. If we go back and look at our sequential mechanism, as we bind an inhibitor, we can actually increase the affinity for more inhibitors, okay, and further decrease the affinity for your substrate. Okay, and that's what they're trying to show with all those figures. So you get a negative cooperativity effect as opposed to a positive cooperativity.
Negative cooperativity meaning just more inhibition and less substrate?
More inhibition, less substrate.
Okay.
The rest of Chapter 7, in my opinion, is actually relatively straightforward. You go through a couple different examples, and they talk about organizing different reactions. Okay? So we can look at phosphorylation as a way to activate an enzyme. Okay? So what we can go through and do, in this case they're referring to a serine residue. We can add a phosphate group to that using ATP, and that now phosphorylation increases the activity of the enzyme. Okay? Why might it increase the activity of the enzyme?
Because
because you have 4 oxygens now instead of
You've
single oxygen.
change the reactivity around that oxygen significantly. Okay? That change in activity is really what's going to facilitate further reactions happening. Okay? Remember, your primary storage of energy is in the phosphate group. So we form ATP to transfer that phosphate group because it changes the energy dynamics significantly. Okay? Why might phosphates affect allosteric reactions? Shape and charge. If we look at the rest, or that structure, if we start with the serine residue, okay, we're looking at a relatively small structure, or small side chain, okay, with just kind of an intermediate polar group. If we now change that to a phosphate, try to emphasize it, the phosphate is significantly larger. There's more atoms. Okay, so we get a size effect. Further, what does the phosphate bring in? Brings in charge. So we're seeing a pretty drastic change in the physical observations by just putting on a phosphate group. Okay? So those subtle changes in the structure can result in a significant change in the structure, the enzyme structure itself. Okay? What amino acids might you expect to be prone to this activity? It's a trickier question. You've got one really easy. Serine. Okay, now you got the easy one.
The acids? These are negatively charged?
Polar uncharged.
The acids are going to be a bit challenging. Okay, why might the acids be a bit tricky to go through and phosphorylate?
You pronate them?
No, we still got that negative oxygen.
Resonance?
Resonance does what to the negative charge?
Stabilizes
Stabilizes it, which makes that oxygen less of a
Acceptor of...
less of a reactive species, fair enough. Less of a nucleophile. Okay. If it's not a good nucleophile, it's not going to react very well with our phosphate group. So our acid groups, while they do have that oxygen, probably not the best. We could do it, but probably not the best. So we're looking for any of our nucleophilic R groups. Serine works, tryptophan, right? Tryptophan, no, I'm thinking tyrosine. Sorry.
Would be all the bases then?
Okay. Your bases could potentially, but again, those aren't going to be great nucleophiles. Some of them would work, like maybe your lysines.
Your polar uncharged.
Okay. You're looking at your polar uncharged ones primarily. Okay. Would they only act as activators? Well, there's really no context to this, whether it's an activator or a deactivator. The only context is that we're saying, hey, phosphorylation can activate. Why can it activate? It changes the structure. Well, does it have to change the structure in favor of running a reaction? No, it can change the structure in favor of deactivating a reaction. I'm not sure I'd go as far as a protecting group, but it would definitely change the dynamics. Yeah. One big term that comes out of this, kinases. Kinases are the group of enzymes that catalyze the transfer of a phosphate group, okay? Which is a question out of the homework, just so you know. So the enzymes that facilitate that phosphate are just referred to as a kinase. Nice and straightforward. Zymogens are another example. Zymogens are an inactive form of your enzyme. Okay, so it's our protein, say that all folded up, but it's now not active at all. Okay, and it's physically inactive, meaning there's something covalently wrong with the structure that prevents it from activating. So chymotrypsin is a nice example of this because it goes through a couple types of reactions, okay, where it cleaves or it gets cleaved at certain locations. Once those covalent bonds are broken, it now allows for the proper folding of the enzyme, and it now is in its active state. Okay, so what they're referencing here is the different breakages. So what we get in this first case is a cleavage by trypsin. So trypsin is an enzyme that comes in and breaks that covalent linkage in the peptide backbone. Okay. What makes this even more interesting is this next statement. What's the next reaction? self-digestion, meaning the now partially activated enzyme attacks itself to cleave yet another bond to then allow it to go through and cleave out some particular sections. And what they're showing by those yellow linkages is now how each of those different protein sequences kind of interact with each other. Okay? So if we take a look at the top, those yellow sequences have not changed all the way through this. What's happening is that an enzyme comes through, does some cleavage for us. Once that cleavage is set up, then our enzyme can self-cleave out a couple sections, and we lose those two pieces, and we now have our fully active enzyme, which can now go out and do all its fun stuff, which we talked about previously. Chymotrypsin can actually go through and cleave, what was it, phenylalanine and tyrosine? I think it was those two residues. Did I put that up there? I didn't. That's off of one of our older slides. Okay. Active site chemistry, now that I'm out of time. We've got all sorts of fun things to consider, which we'll consider on Thursday. You're looking at primary sequence effects, your secondary plus effects, organic mechanisms, so what is physically changing, how are the electrons moving, what types of mechanisms do we have, specificity, okay? Everything in biology is specific due to chirality. Okay, so they give you kind of a neat little picture that shows that. Our enzyme has to have those perfect substrates aligned appropriately, and we'll see chymotrypsin pop up again as an example where we actually look at some bond cleavages.